Introduction to Cleanroom Microbiology. Welcome to this PharmaTalks training chapter on the scientific foundations of contamination control in pharmaceutical cleanrooms. We will connect microorganisms, facility design, operations, monitoring, and patient protection throughout the presentation. Please subscribe to PharmaTalks and explore the other videos in this learning series. Leave a comment below so we can send you the file related to this video, the complete educational file collection, and information about the relevant courses. What this chapter enables. By the end of this chapter, you should be able to explain the main contamination mechanisms and distinguish meaningful evidence of control from superficial signs. You will learn to recognize recurring failure patterns and support decisions that are proportionate to product and process risk. The objective is to understand the concepts, apply them in the operating environment, and verify that the controls remain effective. The chapter control system at a glance. The control logic begins with recognizing a hazard, then identifying its transfer pathway, selecting preventive controls, and collecting evidence of sustained performance. Technical values such as airflow velocity, pressure differential, and cleanup time are useful reference points, but they must be applied within the actual site context. Monitoring alone does not create sterility. It provides evidence about the performance of the preventive control system. Purpose, scope, and patient protection. Cleanroom microbiology links microbial behavior with the design and operation of pharmaceutical controlled environments. The significance of an observation depends on product exposure, process state, exposure duration, and the strength of the existing controls. Every decision should therefore begin with patient protection and the prevention of contamination reaching exposed product or a critical surface. Controls must interrupt the complete pathway. Effective contamination control requires us to examine the source, the transfer route, and the vulnerable target at the same time. A preventive control should interrupt the route before contamination reaches exposed product, a sterile component, or a product contact surface. Confidence increases when independent controls break the pathway at more than one point. Evidence needs context. Failures reveal mechanisms. A count or test result has limited interpretive value unless its location, time, operating condition, and associated intervention are known. A room that looks clean or a sample that shows no recovery is not proof of sterility. Failures should be used to identify the actual contamination mechanism and strengthen prevention, rather than being closed as unrelated isolated events. Regulatory cleanroom technical reference. This table places non-viable particle limits and routine microbial action limits for different cleanroom grades in one technical view. The large differences between grades demonstrate why the level of control must match process criticality and the degree of product exposure. These values should be interpreted with current requirements, site commitments, and trend data, rather than copied without an applicability assessment. Microorganisms relevant to pharmaceutical operations. Vegetative bacteria, bacterial spores, yeasts, and molds differ in their sources, survival characteristics, resistance, and pharmaceutical significance. A low count of an unusual or resistant organism may be more important than a higher count of expected facility flora. Identity, recovery history, sample location, and product susceptibility should always be evaluated together with the final count. Controls must interrupt the complete pathway. Control measures should consider growth requirements, spore formation, resistance to drying, biofilm potential, and whether an organism may be objectionable. Moisture control, controlled material transfer, cleanable design, and disciplined personnel behavior can interrupt survival and transfer routes. Final disinfection alone cannot compensate for conditions that continue to support microbial persistence. Evidence needs context. Failures reveal mechanisms. Microbiological evidence should extend beyond the reported colony count to include identification method, incubation conditions, isolate history, and product susceptibility. Judging every recovery only by CFU can overlook slow-growing, injured, or resistant organisms. Trends in organism identity and recurring locations often reveal the contamination mechanism more effectively than a single result. Contamination hazard characterization. This table distinguishes vegetative cells, spores, yeasts, molds, non-viable particles, and endotoxin by their sources, measurement methods, and warning signals. For each hazard, the control evidence and the condition requiring escalation should be defined in advance. This distinction allows the response to follow the actual mechanism instead of applying one control method to every contamination type. Applied view: microorganisms relevant to pharmaceutical operations. When reviewing this operating image, look beyond the clean appearance of the room and garments. Trace hand contact routes, material movement, airflow, equipment positioning, and the location of critical surfaces. Field observation should confirm that the controls defined on paper also perform as intended under the real operating state. Viable, non-viable, and pyrogen contamination. These hazards may occur together, but they are not equivalent and require different measurement strategies. Particle counting does not identify how many viable microorganisms are present, and the absence of microbial growth does not exclude endotoxin or cellular residues. Risk assessment should consider the product, route of administration, process state, and transfer mechanism for each hazard separately. Controls must interrupt the complete pathway. Viable contamination requires control of microbial sources and recovery conditions, while particle control relies heavily on airflow, equipment, and behavior. Pyrogen control depends on material quality, process design, and effective cleaning or depyrogenation. These data streams should be reviewed together, but one should never be used as a substitute for another. Evidence needs context. Failures reveal mechanisms. A particle is not automatically a microorganism, and microorganisms are not reliably represented by particle counts alone. Likewise, a single test cannot demonstrate every dimension of contamination control. Combining viable data, particle trends, process events, environmental conditions, and behavioral observations provides a more defensible picture of the state of control. Cleanrooms as a system of controls. A cleanroom is not simply a room with polished surfaces and a clean appearance. It is an integrated system of engineering, procedural, and behavioral controls. HVAC. Pressure differentials, barriers, material flow, cleaning, and personnel performance must operate together. Weakness in one layer can reduce the effectiveness of the others and reopen the contamination pathway. Controls must interrupt the complete pathway. A HEPA filter cannot compensate for uncontrolled material transfer, inappropriate contact, or an intervention that blocks first air. Pressure, airflow barriers, and work practices must be designed to prevent contamination moving from a less clean area toward a critical target. The performance of this chain should be demonstrated under representative operating conditions. Evidence needs context. Failures reveal mechanisms. Qualification results, airflow studies, alarm records, preventive maintenance, and direct observation of operator behavior should be reviewed as one evidence package. Excessive reliance on HEPA filtration or failure to assess uncontrolled interventions can create false confidence. Each alarm or deviation is an opportunity to identify the weakened control layer and verify effective recovery. CCS Lifecycle Control Matrix. This table connects lifecycle elements with design inputs, qualification evidence, and triggers for change assessment. Every control should be traceable to its design basis and supported by performance data during routine operation. Changes to rooms, equipment, personnel roles, materials, or cleaning methods should be assessed so the qualified state is preserved. Contamination Control Strategy Fundamentals. The CCS integrates facility, process, personnel, utility, cleaning, monitoring, and investigation controls within one coherent framework. It should not be a simple index of standard operating procedures. It should demonstrate the relationship between hazards, controls, evidence, and accountability. An effective CCS is a living lifecycle system that evolves with changes, trends, investigations, and new knowledge. Controls must interrupt the complete pathway. A strong CCS gives priority to prevention through risk-based design, a hierarchy of controls, meaningful detection capability, governance, and periodic review. Monitoring is most valuable when it can identify declining performance before product protection is lost. CCS decisions should clearly show what protects the product, who owns the control, and which evidence confirms effectiveness. Evidence needs context. Failures reveal mechanisms. The CCS should be supported by documented links to risk assessments, performance indicators, and management review. Listing controls without testing their interactions, distributing ownership too widely, or relying on a fixed annual review can conceal real system weaknesses. Review frequency should respond to changes, trends, and operating signals, not only to the calendar. Regulatory and standards landscape. Regulations, GMP guidance, pharmacopoeial chapters, and ISO standards serve different but complementary purposes. Some establish legal requirements, some describe regulatory expectations, and others provide technical methods for classification or testing. Each site should determine which references apply to its products, markets, and processes, and show how they are implemented within the quality system. Controls must interrupt the complete pathway. EU GMP Annex 1, FDA Aseptic Processing Guidance, whose Sterile Product Guidance ISO 14644 and ICH Q9 each support a different part of the control decision. The goal is not to collect the names of standards, but to convert their expectations into practical controls, justified limits, and reviewable evidence. Ultimately, the control must interrupt contamination before it reaches the product. Evidence needs context, failures reveal mechanisms. Before adopting any numeric value or requirement, confirm the current document version, jurisdiction, process type, and site commitments. Copying limits without context, treating a voluntary standard as complete GMP, or relying on an obsolete addition creates compliance and quality risk. A documented gap assessment should show how each applicable expectation is addressed. QRM scoring inputs for cleanroom hazards. This table shows how product exposure, transfer plausibility, control independence, detectability, and uncertainty influence risk priority. A score is a tool for structuring scientific discussion, not a replacement for specialist judgment. Every score should be supported by defined evidence and should lead to a clear, defensible operational decision. Quality risk management in cleanrooms. QRM connects scientific knowledge with proportionate decisions about control strength, monitoring, and resources. The importance of a hazard depends on product exposure, the probability of contamination reaching the target, the strength of the controls, and the uncertainty in the available information. The assessment output should identify the required action, its owner, and the conditions that trigger reassessment. Controls must interrupt the complete pathway. Hazard identification, severity, probability, detectability, uncertainty, and bias management should all be addressed transparently. A final score is useful only when it shows which contamination pathway must be interrupted and which control will achieve that outcome. If a team produces numbers without a decision or an accountable owner, QRM becomes an administrative exercise. Evidence needs context, failures reveal mechanisms. The quality of a risk assessment depends on the quality of the question, the evidence, and the multidisciplinary team. Data inputs, review triggers, uncertainties, and the decision record should be clearly documented. False precision, biased workshops, and failure to revisit assumptions can make a significant risk appear artificially small. Roles competence in quality culture. Sustained control depends on people who recognize weak signals and act before contamination reaches the product. Having INSOP is not enough. Personnel must understand the scientific reason for each step, their decision boundaries, and the escalation route. A healthy quality culture supports reporting, questioning, and stopping work when product protection may be compromised. Controls must interrupt the complete pathway. Microbiology, engineering, operations, quality assurance, validation, and maintenance each contribute different evidence and responsibilities. When information remains isolated between these functions, even a small change may be implemented without understanding its wider impact. Clear governance ensures that decisions consider the product, facility, process, and microbiological data together. Evidence needs context, failures reveal mechanisms. Attendance at training is not the same as demonstrated competence. Role profiles, curricula, practical qualification, behavioral observation, and escalation performance should show that an individual can execute the control correctly. Attendance-only training. Normalization of deviance and unclear stopwork authority create hidden and recurring failure mechanisms. Cleanroom governance and review cadence. This table clarifies system ownership, controlled records, required review inputs, and expected performance outputs. Quality, microbiology, engineering, operations, and validation should bring their data together within a coordinated review cycle. The review cadence should match risk and the speed of change, while important signals should be escalated immediately rather than waiting for the next scheduled meeting. 6 checks before declaring control. Confirm that scope, patient protection, and the relevant microorganisms have been clearly defined. Then verify the distinctions among viable, non-viable, and pyrogen contamination, the performance of the cleanroom system, the CCS, and the applicable requirements. Each area should have an approved control, an accountable owner, current data, and evidence of effectiveness before control is declared. Control is demonstrated through connected evidence. The central conclusion of this chapter is that the hazard, transfer route, vulnerable target, preventive control, monitoring evidence, investigation, and verified improvement must form one scientific narrative. Prevention has priority, detection provides warning, learning explains the mechanism, and improvement must demonstrate effectiveness. With this approach, a cleanroom becomes a defensible patient protection system rather than simply a clean-looking space.